Thank you very much for your kind invitation to speak today. I think David has done a nice job at covering the SNP side of genome-wide association studies. And as you may know, the microarrays that are used for SNP-based GWAS can also be used for copy number-based genome-wide association studies. And so I have three main sections to my presentation. I'll be talking about experimental design for SNP and CNV GWAS. I'll talk a bit about how we do the calls, how to transform the experimental data into copy number information, and then go on to talking the main bulk of the presentation, talking about the quality control facets, and then the actual how to find the relationship between copy number data and phenotypic information. So the first section is very near and dear to my heart because we've done so many studies and found that there have been experimental design problems that cause many challenges in quality control and association analysis. And when I talk about the design of experiments, there's the facet that David spoke about, which is around sample size and whether you're going to do a family-based study or a case control-based study. But the issues of the experimental design that I'm talking about, maybe we could almost say the design of the actual experimental protocol, namely issues like cases and controls not being balanced in the plates. So typically they run 96 samples on a plate or 48 samples on a plate. And if you do all your cases on one set of plates and all your controls on another, the experimental difference between those will cause spurious associations that are a function of the experimental difference from plate to plate. This happens also when we do things like try to do meta-analysis, combining studies, particularly when you use different microarray platforms. Splitting trios or families up is a particularly troublesome way to do the study. In other words, you want to make sure that your entire family unit is on the same plate and run at the same time. So what happens if you run into these challenges? There's endless struggles with type 1 error. Often we're discarding large fractions of the genotypes in SNP-based studies. Copy number studies actually, because you often cannot make a clear discrete copy number call from a single probe like you can do for a SNP. Copy number studies, experimental design is even a bigger issue, probably order of magnitude bigger of an issue than with SNP studies, where you can to some degree get away with these issues I've talked about. So to give you a little picture of this is the Wellcome Trust Type 1 Diabetes SNP study, and we did this principal components analysis looking at the first two components on the SNPs, and we've also did it for the GAIN study. Now the Wellcome Trust study, as was described by David, they ran the cases and the controls separately. Actually, I don't know if it was obvious, but they were run in entirely different experimental batches. And the first two components actually don't show much of a difference, and this is actually just population structure we're looking at. So indeed, they're mostly drawn from a European population. However, when you do an analogous principal components analysis on the copy number intensity data, the log ratios, you see this large shift between the case and control populations as well as some major subgroups. We've often seen things like the scanner that's used can make a big difference. You'll see this whole cluster of individuals who are run on one scanner versus another scanner. And I'll talk about how to address this in your experimental design later, but if you don't take this into account, you're just going to have a lot of grief downstream. Now, we tend to some of the things that could have been very embarrassing if we were to publish and say, aha, we've got a finding, is exemplified right here. We had a 4-site study, so there was actually 4 different places the genotypes were collected, and they were actually run on at different times experimentally. The blue is a set of cases. It was about half of the samples. The other 3 colors in the histogram are different control samples. And so when we ran an association test, we saw a strong, this is the histogram of the intensities for a particular probe somewhere in the genome, and we saw a large difference between cases and controls, mainly driven by this yellow set of controls from site number 1. And whereas you see the cases are centered here, and there's this bimodal kind of distribution for controls. If we were to conclude, aha, there's some biological difference here, there would have been egg on our face, because if we hadn't run these 2 other controls that actually ended up having the same mean as the cases, we wouldn't have suspected this batch from site 1 in yellow as being different. And in fact, when we looked at the intensity data and so forth, we could see that this was just, the probe behaved totally differently for this set of experiments. We're not sure what was different about the experimental conditions, but it was driving the association rather than some biological effect. So how then do we minimize these sources of variability? And paying attention to this as you're working on your study makes everything after this go much easier. So the things that cause variation, plate-to-plate variability seems to be the largest. And we'll talk about designing a plate design to account for this. It could be due to how the DNA is amplified, perhaps how long the hybridization process goes, what reagents are used, different scanners. Environmental variability, such as temperature, maybe temperature's not such a big deal, not sure. Ozone can be a big deal. There's a paper by the folks at Signature Genomic Laboratories. They found that the quality of their data, and they were running array CGH chips, Agilent array CGH chips, the quality of their data varied as a function of the train schedule of the nearby train station. Their lab was very close to a train station, and the ozone produced by the trains actually was having a measurable impact on the quality of their data. So they put ozone hood over their instruments, and eventually they moved their lab away from the train station, but and then still had an ozone hood and on top of an overall HVAC system that could keep ozone levels low. So DNA concentration seems to be a culprit in causing a wave effect phenomena, where you see this in this green line. There's these wave effects. So it's not actual gains or loss in copy number, but rather, again, an experimental artifact. There's been some advances in trying to correct this stuff, correct these wave effects mathematically. But there's always problems when you're trying to sort of fix these errors that are introduced. Also, be aware that differences in blood, in the source of blood, we've run a lot of HapMap samples as controls and regretted it because they came from cell lines, and they often behave very differently than the blood-based DNA that was just extracted from blood and not gone through an immortalization process. So one of the 270 HapMap samples that are public, that everybody downloads and does all sorts of analysis with, if you look at the copy number intensity for NA18540, there's 5 chromosomes that have an extra copy. Now, and this is a healthy individual, so there's no way somebody would even be born with extra copies of 5 chromosomes. It's clearly a cell line immortalization artifact. So we recommend solve this problem at the beginning. essentially look for any source of variability and try to be consistent. Extract the DNA from the same site, the same source, use the same extraction kit, the same time between freezing, collect your cases and controls at the same time if at all possible. And again, whole blood, generally we would prefer that. Saliva, if you're going to do, say, saliva, do it for everybody, your cases and your controls. Plating, and we'll talk later, use design of experiment principles to randomize your phenotypes across the plates, and I'll talk at length about that a little later. We recommend placing a well-characterized sample, maybe even two, a male and a female, on every plate, so you have a consistently the same sample from plate to plate, so you can really look at the variability. Potentially even use it for normalization and correction of distortions due to variation from plate to plate. We ran this large study, as I mentioned earlier, where we used HapMap samples as those two, and we regretted it because they were cell lines. So actually it'd be best if you just choose one or two of your own samples and just draw a lot of blood from them and extract a lot of DNA and use that for control. So I'm talking about experimental control as opposed to a case control. Keep families together. Do not leave it up to your core lab to hope that they will randomize for you. They won't, and you'll regret it. Buy all your arrays in a single lot. Run your samples together. There's many. We want to be, share in the experimental credit sometimes, and we'll run a study at multiple genotyping centers to kind of maybe split the credit for doing it or something. It's not usually a good idea if you can help it. Do it at one location, so you're minimizing the variability of using entirely different genotyping centers. And then often, as I'll talk about in the QC section of the presentation, the typical quality metrics that are done at core labs just look at genotyping quality and a little bit looking at things like the derivative log ratio spread, which is a measure of the noise in the data. They're not always sufficient to capture the problems that you will see in CNV studies. So sometimes a SNP study you would keep 95% of the samples, and then you would move them on to a CNV study and discard another 20-30% that had problems with wave effects, noisiness, and so forth. And so if you're going to rerun your samples, it's best not to wait 5 months or a year later to discover you have quality problems, but to be able to do that analysis early. And it's as I said, 95% of the studies we've looked at, these have come from major centers, the Broad Institute, Harvard, and then the little centers as well, have had these major problems. It's changing, but if you're beginning to do your own studies, hopefully you can avoid this pain by taking this advice. Okay, so say a bit about, I think it's been talked about nicely by David about family versus population. One extra thing I'd add about family studies, they're very nice under the rare variant hypothesis if you have a large family, right? So if you've got a large pedigree, you've got a rare condition, you can potentially track it through the entire pedigree. And the nice thing is you can also figure out if there's de novo copy number changes, something that where the two parents do not have the copy number change, but the offspring might. And in autism, it's apparently something like 7% of the autism cases. It looks like there's de novo copy number variants. So the downside of family studies is as David said, you have to capture more individuals for the same power. And it's not always easy for late onset diseases to capture the parents or better yet, even grandparents.